will be using microscopes today. So, oh yes, and this is from Aurora. Thank you, Aurora. Yeah, we'll be using microscopes today. I think so too, Jaden. Yeah. And the reason is because when we get back from spring break, we are starting the biology unit. So our learning or learning objective is to conduct an investigation to provide evidence that living things are made of cells, either one cell or many different numbers and types of cells. So a microscope is a great tool to be able to conduct that investigation. So today we are introducing ourselves to the microscope. Some of you guys have already used microscopes, so it's less of an introduction, but we're going to be walking through the steps of setting up the microscope, cleaning up from a lab, and we will be doing a lab today as well, so that's pretty fun. So our two questions that we're going to think about today as we complete our activities and then also as we move through the unit is how do we define something as alive, and how has the microscope changed our view of ourselves in the world. So on the front or on the middle of your tables, I put a worksheet. This is a video that gives us not just an introduction to microscopes, but also tells us a little bit about conducting this investigation. So there are a few of these that they answer back to back. So just do your best to write in the responses, and then we'll go over ones that you missed at the end. Okay, so take a second, read the questions that you have on here so you kind of know what to look for. On the front is what we're going to complete together with the video. Yeah.
She got the answer sheet.
No, those are just sentence starters.
Yep, it's just sentence starters.
To help you fill in as quickly as possible.
Are you telling me I'm a slow writer?
I am.
Wow.
Okay, so take a second to read through there, and then we will watch the video.
No offense. I just want to make sure you get all the information.
Oh, that's good to highlight some.
You're too busy playing with your hair.
Okay, here we go.
Right, well, look, you didn't write your name. Oh, case in point.
No.
What is on the kitchen table? Oh, it's my roses. I'm hoping to find some Euglena, maybe some Arancia. I took the sample from and then why is it on the kitchen table? Oh, I got distracted. I was gonna take some drops and put it on the slide while I set my microscope. Yes. Well, please get your green water away from where we eat. Green water? Don't be ridiculous, Petunia. There are tons of protists in here. We just have to find them. Well, you're out of luck. The protists that I'm looking for are unicellular, meaning they're made up of one cell. And generally, you need a microscope to see them. Never used one of those. You've never used a microscope before? No. Well, you know what that means. I'm afraid of what that means. We must explore the world of microscopes. First of all, let's focus on a few terms that are important in order to understand microscopes. Magnification is one. That's really what most people think of when they think of a microscope. And we're going to talk more about that in a moment. But another term that is important is resolution. Basically, are you able to distinguish between two objects? This is important as otherwise two objects could actually look like one object because you can't distinguish between them. So for example, if a microscope had a resolution of 0.2 microns, then that means two objects need to be 0.2 microns or more apart in order to see that they are two different objects and not one. If you want to understand the physics of how resolution works and how it's calculated, we have a great further reading suggestion in the video details. So let's talk about some different types of microscopes that all take into account this need for magnification and resolution in order to see specimens. First, light microscopes. If you're in a classroom, this is likely what you have. And as its name would suggest, it uses light in order to see an image. Bright field light microscopes tend to be what people are most familiar with, typically producing a darker image on a light background. With a dark field light microscope, you have a piece that blocks the light source, called a light stop. The idea is that most light has been blocked, so the only light you see is light reflected or refracted from structures within the specimen that you're looking at. So you tend to see light images on a dark background. There are many other types of light microscopes too. We have a great link for further reading suggestions to learn how each of these work.
And some of these are fascinating. Some rely on interference, some rely on using a laser, but the thing to keep in mind is there are certain times when one of these types of light microscopes is more ideal to use than another. So light microscopes use light in some form to see a specimen. But not all microscopes are light microscopes. For example, what if we wanted to see a virus? Viruses are generally very small, typically much smaller than the average bacterium. How can we increase magnification and resolution abilities to That's where electron microscopes come in. Electron microscopes use electron beams to visualize images. Transmission electron microscopes, TEMs, tend to be the most ideal for visualizing structures within a specimen, while scanning electron microscopes, SEMs, tend to be most ideal for visualizing the 3D surface. Let's come back to the typical microscope you might see in a classroom, which happens to be the one that I own, a bright field light microscope. Here's our light source. Light passes through this, the condenser lens, to focus the light on the specimen. The level of light can be adjusted by the diagram. Our specimen, likely to be on a microscope slide, will be placed on the stage here. Light will be reflected, diffracted, or absorbed by the specimen. But we rely on these lenses here, the objective lens and the eyepiece lens, to get our total magnification. In fact, this is often referred to as a compound microscope because it has two sets of lenses. You can see that the objective lenses are attached to this revolving nose piece, which allows them to be rotated. So you can select a certain one that you want to use. With my particular microscope, I have 3 objective lenses. A scanning objective lens that magnifies 4 times, the low power objective lens that magnifies 10 times, and the high power objective lens which magnifies 40 times. But don't forget the eyepiece lens, because that also magnifies images. And my particular eyepiece lens magnifies 10 times. So let's say I used the scanning objective lens. I would multiply that objective lens magnification level by the eyepiece lens magnification level so that my total magnification is 40 times. Now you'll also notice there's a lot of knobs here. Let me introduce you to the knobs. This large knob here is known as the coarse focus, and then this smaller knob here is known as the fine focus. Both of these knobs raise or lower the stage, which helps with focusing. The coarse focus knob will move it more dramatically, whereas the fine focus knob will move it in smaller increments. These knobs, the stage knobs, don't move the stage up and down, but rather from side to side instead. It helps you explore what's on the slide itself, but it isn't adjusting the focus of it. Not all microscopes have those stage knobs. Sometimes you have to move the slide manually. This is the microscope arm and the base. Whenever you pick it up, you want to make sure you have one hand underneath, supporting the base, and the other hand holding the microscope arm. Since it plugs in, you want to make sure there isn't water around it. So let's take a look at what's in this water sample here. I'm going to do something called a wet mount to prepare my microscope slide. I'm going to drop a drop or two of my water sample onto the slide using a disposable pipette. It's like a very fancy eyedropper, kind of. I'm going to put a cover slip on top. There's some techniques to doing a wet mount to avoid air bubbles that you can check out online. Air bubbles actually can look kind of beautiful under the microscope, which can lead to great disappointment when you find out they're not some amazing microscopic organism, but rather just an air bubble. Something I may or may not know by experience. I'm going to put the slide on the stage. I'm going to use the stage clips here to secure the slide. Light is on, and I already have the scanning objective lens here ready to go, which is good because I want to find the specimen first. I can adjust the light level if I want, but just know that super bright doesn't always mean the best image. I can look through the eyepiece and use the coarse focus knob to move the stage up or down slightly to focus. Patience. Euclina. I can also fine-tune focus it with the fine focus knob. I can use the stage knobs to center my image. Now that I've found it with the scanning objective lens, I'm now going to move up to the next magnification level. So remember, total magnification would be 100 times. Since I'd already focused it with the scanning objective lens, it's likely I only need to use the fine focus knob to adjust the focus. I could continue to move up to the high power objective lens, which would give me a total magnification of 400 times. Now, 3 additional tips about using microscopes that I just want to mention. Number 1, most microscope slides are glass, so they can break if dropped. And if using cover slips, keep in mind they are extremely easy to lose if not careful. Number 2, always be aware of where the slide is when you're moving the stage up and down with the focus knobs. It's possible to move the stage too close to the objective lens and actually crush the slide. And number 3, if it's hard to see your image even when focusing, you may need to clean the lens. But you need to do this by wiping it with lens paper, not regular tissue. When you're done with the microscope, make sure the slide is removed, turn off the light, lower the stage to its lowest position, and return the lowest objective lens over the stage. Unplug it, wrap up the power cord, and if you have a cover the microscope before putting it away. Keep in mind there are some techniques to better visualize some specimens, such as using different stains or using immersion oil at highest magnification to increase resolution. Definitely something to explore that this video doesn't go into. Microscopes open up an entire world that you can't see with the naked eye. Microscopes can also complement what you might be learning in life science. Studying mitosis? Check out the cross-section of an onion root tip where lots of mitosis happens. Studying plant responses? Take a look at stomata from a pen sample underneath a leaf. Studying osmosis? Take a look at how aquatic plant cells respond to different salt concentrations. Endless possibilities to explore endless curiosity. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay. So, okay. Anyway, let's go over a couple of these questions before we move on, because I know some of it was, back to back was like rapid information. So just to hit on some of these that are important for us right now. The first one asks us to explain why both magnification and resolution are important to seeing a microscopic organism. So what did you guys get? What is magnification and resolution? What is magnification? If I magnify something, what am I doing?
Zooming in.
What's that?
Like zooming in.
Yeah, zooming in, making it bigger, right? So magnification makes your image bigger, right? It enlarges your image. So that is important when we are trying to look at a microscopic organism, because otherwise you're not going to see it. You're not going to be able to see it. So magnification makes your image bigger, okay? And then resolution. So resolution is a little bit different. What is resolution? Somebody got it. Yeah, Maya.
You like distinguish the thing.
Okay, Cabs, you want to help her?
Resolution makes things easier to see. It makes it clear.
Yes, exactly. Resolution clarifies your image, so it makes it clear. So what we're going to see in our lab today is that once we get our sample focused, as soon as we go to the next magnification, it's blurry again. So we may have magnified it, but now we have to use resolution to bring it back into clarity, right? So we have magnification makes an image appear larger, and resolution allows you to see it clearly. Okay, so that should be what you have for number 1, are those two things. Number 2, real easy difference between a light microscope and electron microscope would be what? What does a light microscope use to be able to see? Light. What does an electron microscope use? Yeah, electron beams, right? So that would be your major difference between the two. Now the video went in to talk about how electron microscopes kind of give us more detail. For what we're doing in our unit, the light microscope is going to be perfectly fine to be able to see and investigate the things that we want to look at. I'm going to skip number 3 for right now, but let's go to number 4. So there were 3 objective lenses that were mentioned, 4x, 10x, and 40x. What were those called? Do you guys remember? Did anybody write it down, what they were called? Maya?
With like the ID.
Yeah.
40, 10, and then 40.
Oh, okay, yeah, so down, are you talking about the scanning objective times the eyepiece? Okay, yeah, so when we look at the objective lens, it might tell us 4x, 10x, or 40x, but we have to remember that our eyepiece also gives us a 10x magnification, so it's actually 10 times what it tells you on the objective lens. So that's what you would get there. Did anybody write down what they're actually called? Megan has them written down. What did you put?
Scanning for 4x, low power for 10x, and high power for
Yeah.
10x.
Yeah, so the 4x lens, and we're going to see this on our microscope, is the scanning objective lens, and that's the one you use to actually find your image, that you put it in, center your image, and all of that, and then as you go up, you're magnifying it, right? So we have the scanning objective lens, and then low power and high power. Okay? For the tips, why is it important to remember that the slides and cover slips are made of glass? Yeah. Yeah, they can break easily, definitely. And as we go to the next two tips, we're gonna see why that's important too, to understand. So when using a light microscope, why is it important to use the scanning objective first? The scanning objective lens before you go all the way to 400x. Yeah, gaps. Okay, yeah, first of all, because the scanning objective lens is going to give you the whole picture, right? Because as we said, as you go closer and closer, as you magnify it, you see less of, as far as the surface area of your specimen. So yeah, you want to be able to start there so you can focus it. And then that goes along too with number 7. It says as you move up the high power objective lens, avoid using the coarse focus knob. What was the reason for that? It was a safety issue. So it goes back to those slides being glass. If the coarse focus knob is going to move the stage up and down a lot. So if I move it up, and I move it a lot, and then I go to 100 magnification, it's longer and it's gonna just crush that slide. Okay, so it's for safety reasons too. So you wanna make sure, once you find your specimen, and we'll talk about this when we do our lab, once you find it with the 4X, you shouldn't need to move the coarse knob. You should only need to use the fine focus knob. Okay, so that was part of that. All right, quick true and false on the back. Follow the procedure for a wet mount to avoid air bubbles because they can be mistaken for a specimen. True or false? True, right? We want to avoid air bubbles, and we'll look at that in a minute. The pointer that you see in view can typically be rotated by rotating the eyepiece. True or false? That was true. That was true. You will not have one on your microscope though. If the lens needs to be cleaned, do we use tissue paper?
No.
False. Yeah, we don't want to use tissue paper on the lens. There's special paper that we can use. The light microscope drawn above is also known as a compound microscope as it has more than one set of lenses. True. Yeah. And then oil immersion can be used at all magnification levels. False. That's right. Only at the highest. Okay, so you guys can put that paper away. And let's look at our lab. Thank you. Okay, so what's going to happen is you will work with your table partner at a microscope to complete the lab. So the lab papers are up at the microscopes for you. Also, the materials you need to make your slide for today. So you have slide materials, you have your lab, and then you have your microscope. So what we're going to do is you and your partner, you're going to find a microscope, and you're going to go up there and read through your lab really quickly so you understand what we're going to be doing today. And then I will direct you to creating your slide. Mr. Eller or I will come by with some water to be able to help you create your slide. So once you get up there and read through your lab, you're going to complete numbers 1 and 3 with your partner while you wait for your slide to be created. Okay? Yes? Okay, so, since it's a smaller group, we'll just do like whatever microscope you're closest to, go for it. Shannon, do you wanna work with Jaden? Or work over here
As like a partner? Is it like a partner?
as a partner? Yeah, is that good?
Yeah.
Shannon, are you good? Okay, so find a microscope with your partner, go ahead and start reading through the lab, and your answer numbers 1 through 3.
Sebastian, come over here. Get on you over here.
Oh, you guys can, you know, here, my estimate.
10 tough moments. I'm gonna go first.
Does everybody add a microscope? We're all set? Okay. All right, great.
Guys,
Arm and Rain. Okay,
the little kid.
Make sure you have your slide ready. Take the E and the slide cover off until we put the water on.
And you can take the slide cover off.
So once I put the water on, I want you to so that even when you put it on, so just hold it now. So you have to drop, so you're gonna put your E on top and then the cover slip. And this is not too worry too much about air bubbles from this one.
There we go.
You should put it on and then 45 degrees. Or fix that so it's even. And then so your cover slip. Okay, hold on.
Do we plug it in now?
So, you're going to put it on like this, so that you can read it. Good to go?
I see that.
Okay. Let's do this. Okay, so now it doesn't have to be centered, but you want to put it
I see. There's nothing on there. There isn't a slide.
ladies, I'm just gonna give you.
Let me see. Let me see. You can
And
see
then what you're going to do is you want your E to face you like this, so you can read it through just sitting like this. So you put your E on top and then the cover slips.
Wait, if you guys take this?
No, so that you can read it.
those black
I'm
dots.
looking at it like this.
It's not bacteria. Then what is it?
No, right?
Always the material.
Ladies? Okay, so here goes your E slide. So let's see. So once I put water on there.
Alright, so take your slide, bring it closer to the
Just like this.
your slide. Okay,
Put
good.
your cover on.
Okay. And so you're going to
Okay.
take your E. Alright, put it on top of the water droplet, and then you're going to take the cover slip. You're going to kind of angle it at a 45-degree angle and let it fall down so that you don't have any, or try to not have any water bubbles. Okay? So make sure the E is kind of like you read.
Okay, I'm just gonna give you another minute or two to finish reading through.
Then do it.
And answering questions.
Flip it around. No, he said 45-degree angle.
Alright,
Alright,
nobody
so-
should be playing with this.
Yeah, don't mess with the microscopes yet.
There we go.
Wait until she gets instructions from
Okay. Okay, so if I can have your attention up here, this is just a short, maybe 1-minute video to show you the different parts of your particular microscope as we set it up, and then we'll walk through it together. So we'll use this one as an example.
Retrieve a microscope from the cabinet. Carry the microscope with both hands, one holding the arm and the other under the base of the instrument. If the microscope is too heavy for you to carry safely, ask for assistance. Unwind the cord from the back of the microscope and plug it in. The on-off switch is on the back of the scope and may be hard to find if the power cord is still wrapped around the
yours
holders.
is on top.
Check the ocular lenses and turn them until the number 64 lines up with the white line below the rotating part of the lens. Check that the condenser lens is just below the stage. Put the objective lens on low power and move the stage to its highest position. Adjust the light to the medium or medium-high range. You can adjust the light later to improve the image. The brightest light doesn't necessarily result in the best image. Now you should be ready to start using the microscope.
Okay, so what I'd like for everybody to do is you're going to take the cover off your microscope and you're going to find the closest outlet, power outlet. Gently unwrap your power cord and go ahead and plug in your microscope. Turn it on and your plug is right here, or I'm sorry, your switch is right here on the top of the base. Okay, so go ahead and turn yours on. So let me make sure everybody is able to turn theirs on. Use all your mic, Hannon.
Yep.
Okay, does everybody have light? Yep. We have light, okay. Okay, so right below the switch for the light is the adjustment for your light. So like the video said, you don't want it at its brightest, but you may have to make some adjustments as you go through. So that knob right below the switch is going to help you do that, okay? All right, now everybody, do me a favor and touch this clip right here, this clamp. Everybody do this, the clamp. Yes.
I got it. You guys keep going.
Everybody look at this clamp. So this opens your stage so that you can put your slide in. So everybody put your slide on your stage while you hold this open. And once you put your slide on, that is going to hold it. You release it and that will hold it in place. Make sure that when you are looking at your microscope, you can read the E. So the E should be facing you as you open up